Hello, everybody. This is Yajay here from Heightening Awareness. I decided today that I would take a little bit of time to talk to you about the current coronavirus outbreak and what exactly is going on there. Because I see a lot of people who are very concerned and throwing around all these theories of where it came from and what's going to happen. And I thought it would be really important in order to elucidate and educate you on what exactly is going on. So first, I'm going to talk a little bit about what the coronavirus actually is, and then we'll talk about where it may have come from, why it's dangerous, and what we can do to protect ourselves against the current outbreak. So the coronavirus is a member of the genus Coronaviridae in the larger order of nidoviruses. Now, here I have a picture of the coronavirus underneath a microscope. You will see that it has a sort of fringe or crown around it. This is actually where we get the name Corona from. It doesn't have anything to do with the beer. The beer itself actually likely took its name from the same sort of source that we then got the word Corona from when we refer to this crown that we see outside of the virus. Now, what the coronavirus actually is a single-stranded positive sense RNA enveloped virus. Now, you don't really totally need to understand what all of that means, but from a scientific and epidemiological perspective, it does give us a bit of information in terms of where this virus might have come from and also how it behaves inside of the body. So here is a simplified image of what this virus looks like. You'll see that it has a lipid envelope on the outside that's covered with all of these spiked glycoproteins, and that the genome or the RNA of the virus is on the inside of the envelope in this helical formation with nucleocapsid proteins. Now, we need to understand a little bit about how this virus actually infects us and gets into the cell and what it does. Many of you are familiar with the idea that the virus needs to hijack some of the mechanisms and machinery inside of the cell in order to replicate itself, and that it can't survive on its own. This is one of the indicative things of a virus versus a cell. So here is again, very simplified schematic of a coronavirus attaching to the outside of the cell, coming inside of the cell, uncoding and releasing its genome, and then that genome becomes translated and transcribed into all of the various different proteins that are needed, sent into the endoplasmic reticulum to the Golgi and outside of the cell again. The important thing to recognize here about all of this is really the attachment and entry of the cell via these spike proteins on the outside of the envelope. Now, there are all sorts of coronaviruses that exist. Many people are familiar with the 2003 SARS outbreak, particularly for those who live in Canada, especially in the area of Toronto. SARS stands for Severe Acute Respiratory Syndrome. Now, the thing to take away from this is that acute respiratory syndrome is actually a technical term for the common cold. Coronaviruses themselves are present in the human population all the time, and when you get a cold or catch what we call the cold or getting the common cold, there's a very good chance that you're actually infected with a strain of coronavirus. Now, usually when you get a cold, more than half of all colds are caused by what's called a rhinovirus, rhino meaning nose. And the rhinoviruses actually belong to what's called picornaviruses or picornaviridae. This is the same group that poliovirus belongs to and that other enteroviruses belong to. But about 30% of all common colds are actually caused by forms of the coronavirus. So this version that we're seeing now is essentially just a very virulent evolution of the virus. It's a form that has developed the ability to cause a more severe version of what it already does, when people get the cold and they're sick and they have a fever, and maybe even if they're vomiting a little bit, they might even go, "Oh, I have the flu, I am so sick." But there's a good chance that this is actually just a very severe cold and that it is caused by the coronavirus or a number of other viruses that can cause colds as well, that are in that remaining sort of 20%. But the SARS virus, the COVID-19 virus, and other ones like them are just a little bit more difficult for us to fight off. With our immune systems, they're capable of doing a lot more damage to the body before we're able to make ourselves healthy. And we're going to talk a little bit about why that probably is and what's really going on there. So I mentioned before that there are these spike proteins on the surface of the virus envelope that allow it to attach to the cell and enter. These are very much part of the same system or same class of molecules on the virus that our body recognizes as antigens. It makes antibodies to go and attack and attach to. So when you get a virus, your body either has never seen it before, and then there's a delayed response in order to produce the appropriate antibodies to then really go and neutralize it, or you may have been exposed to that virus or a similar virus, in which case your body is able to respond much faster. In the case of the SARS virus and the current coronavirus outbreak, the reason why it is spreading so fast and the reason why it is potentially dangerous is sort of twofold. The first one being that the spike proteins and other potentially antigenic pieces of the virus have mutated and have developed and have become a version that allows them to still attach to human cells and enter the cell and do its thing, but at the same time are different enough from coronaviruses our bodies are already exposed to that we don't have a way of immediately recognizing this virus, and there's a lag phase in our immune system before it's able to catch up and give us a really good response to take this thing out. Now, the other reason why the virus is likely spreading quite quickly is that it has a very long incubation period. Now, what this means is that this is the period of time where one can actually carry the virus before any symptoms present and one is able to actually identify that they're sick. The coronavirus that we're currently dealing with has an incubation period of up to 12 days. Now, this is significantly longer than the incubation period for influenza. This is significantly longer than the incubation period for most rhinoviruses, and in both cases, that's usually anywhere from 48 to 72 hours after exposure. So there aren't that many people in that time that you're probably going to pass the virus on to before realizing that you're sick and contagious. In the case of the current outbreak, that's almost a 2-week period in which you can be walking around, talking to people, coughing on them, sneezing, etc., before you even realize that the virus is in your body and that you need to take measures in order to stop the spread. Now, we're going to talk a little bit about how the SARS virus or how the COVID-19 virus actually was able to develop these mutations that made it possible for it to get by our immune system. And we're going to talk a little bit about the potential sources for this virus. Now, we believe that the virus occurred and developed as a sort of recombination event, and that SARS was something similar. Here is a picture of the phylogenetic tree of coronaviruses. You'll see that there are 3 different major groups in the coronavirus phylogenetic tree. Group 1 and 2 belong to the human coronaviruses. This is where the coronaviruses that infect us reside. And group 3 is mostly avian coronavirus, the one that comes from birds. Now, SARS was a little bit unique in that it lacked the HE protein that many of the group 2 human viruses have, which makes us think that it came about as a result of some sort of recombination event with group 3. So you might be asking yourself, what is a recombination event? How does this happen? And how does that lead to the production of this new, very virulent, very pathogenic virus? So the best example that I could talk about here is the flu. If everybody really thinks a little bit, it won't take them long to remember H1N1A, the swine flu that hit us a while back. This is a really, really good case for examining how recombination events might occur inside of a single animal that is infected by two viruses. So what really went on there with the swine flu is that a form of influenza from birds that was also capable of attaching to pig cells and infecting pigs got into a pig, and then a human virus, a human flu, that was, again, it was also capable of infecting pigs, got into the pig, and somewhere inside of this pig, there were cells that contained both of these viruses, and both of these viruses were replicating and making their proteins and packaging themselves and doing all that. And you can think back to the image that I showed you before of a coronavirus doing something similar inside of a cell. Now, what would have happened is that pieces of one virus would have mixed and matched with the other and then combined and formed a virus that was sort of combinations of both. As a result, possessing the infectious properties of a human virus, being able to infect and cause disease in humans, but at the same time possessing all these new antigens and things that were brought over from the bird flu that were not necessarily recognized by the human immune system and was a little bit more difficult to fight off than a version of the flu you've seen before. Now, this happens with the flu all the time, constantly. This happens between multiple human flu viruses and occasionally happens between trans-species flu viruses combining in an animal and then reinfecting, all this kind of stuff. This is why it's really important to get a flu vaccine every single year if you're worried about infection, because the flu virus is constantly recombining in this way and producing new versions that are more difficult for our body to fight off. Now, in the case of the flu, this is able to happen so quickly because the flu has 8 different pieces of its RNA inside the virus, meaning that it's possible for 8 from one virus over here to mix up with 8 from another virus over here, and as long as each virus has A, B, C, D, E, F, G, H in those functional groups, whether it's human A or bird A or swine A or whatever, as long as it has the right combination of those 8 pieces, it's able to recombine and leave the cell as a new virus. Now, the coronavirus doesn't necessarily do that, as it only has one strand of RNA in its genome. However, coronaviruses have the largest RNA genome of any known RNA virus. That's just really, really, really big. It's got lots of different A's, T's, C's, all that kind of stuff in its RNA, for those of you who are familiar with how DNA and RNA work. Now, the thing here to consider is that DNA and RNA replication are not perfect, and that whenever you replicate or transcribe or translate DNA and RNA, there's always a potential for error. This happens in us. This is how we're capable of mutating a little bit from one generation to the next and evolving, and this is constantly happening all the time. But when you have a virus that has a very large genome, that means that it has a larger number of opportunities to make a mistake or to mutate and develop. So it could very well be that simply because of the size of the coronavirus genome, this allows the virus more opportunity to make changes within itself, particularly if it is in the presence of other pieces of coronavirus DNA from other strains that are maybe damaged. This is where random recombination might be able to occur between broken pieces of RNA. However, this is quite uncommon. And this could explain where some of these viruses came from. So people are familiar with the SARS, and people remember SARS. That was a really big thing back in 2003. We are pretty confident that SARS developed from a mammal, very likely a civet or some other kind of Chinese ferret badger, whatever it might have been. This is where we think SARS came from. And it's, of course, was localized and came out of China. Now, we need to remember that it's very common in certain parts of China to be in very close contact with a large number of animals, whether it's because you're eating them or you're selling them, or I believe there are certain types of traditional Chinese medicine that use pieces of various species of animals as part of the medication. So being in close contact with the carriers of this virus is very common in that part of the world. This is very much why the SARS came from where it did. Now, there is also something called Middle Eastern Respiratory Syndrome, and this was a bit of an epidemic that happened in the Middle East in 2012. Not everybody is aware of this, but this is another example of a coronavirus that sort of came from animals in a mutated form back into humans in a way that our immune system wasn't super good at recognizing. People believe that this is likely have come from a bat, like a tomb bat or something else, but there was also evidence that suggested that this virus was present in camels in Egypt, and that this is where the MERS, MERS, Middle Eastern Respiratory Syndrome, really developed and came from. So this sort of process of a disease going into an animal or coming and then back to humans, or starting in an animal and then coming to humans, is what we call a zoonotic disease. This is what the current coronavirus is. A lot of people think that it was produced in a laboratory or made by human beings. This is not the case. Just because a virus shows up randomly and is extremely deadly and goes across the world, and it happens to happen at a time when people are during political unrest, does not mean that it is a man-made weapon. If we think back to the epidemic of the Spanish flu that happened in the very early 20th century, this spread across the planet and killed millions and millions of people. It was absolutely horrible, and it really got our society, in the contemporary world, to wake up to the reality of these dangers. And that was a zoonotic disease. That is why it spreads quickly. That is why it is so dangerous, because these mixing and matching of things in other animals from various different viruses, the ability to evolve in these different viruses and make mutations, allows the virus to become something that infects humans but is not very easily recognized by the immune system. And that's basically all you need for something to start spreading very quickly. Now, the current coronavirus outbreak has been suspected to have come from a bat species that is present in China. There was a genomic analysis that occurred back in January that found an incredible amount of similarity between this bat coronavirus and the current coronavirus outbreak. So for those who think that it came from seafood or from a seafood market, it would have only been brought to that market first by a carrier, someone who was potentially exposed to one of these bats, or who maybe they could have eaten a bat. I'm not entirely sure, because I don't live in China and I am a Caucasian person who does not necessarily know how that stuff works over there. But from a biological perspective, we're quite certain that it came from a bat. Now, I'm going to end the video a little bit here, just for the sake of length and timing, and I will make another one where we talk more about what it is that we can do to protect ourselves from this current outbreak.